the title of this one, this is, I think, published in the New York Times. Trillions upon trillions of viruses fall from the sky each day. And to the non-biologist, that might freak some people out, but just remember, trillions and trillions of viruses do not care about you at all. They have nothing to do with you. And so I know with the COVID-19 pandemic, that's really created a lot of angst and fear regarding viruses, but part of what I want to do in this part 2 slide set is to demonstrate that they're absolutely necessary for life on Earth, and in a good way, not necessarily focusing on all the tremendous harm they can do, at least a few key ones, but the trillions upon trillions, most of them do not do anything negative. Another reading I'd like you to look at, both of these readings are quite short, by the way. This is another reading in the video section on what they're thinking about with regard to how did viruses come about, how did they evolve to begin with. And these are the 3 leading ideas, where you have the reductive origin, and then the intracellular origin, and then finally an independent origin. So these are still up for possibilities. There are no fossils of viruses. You can't do that. So it's a lot of speculation. And then there's some scientists who say that it could be multiple mechanisms in play here that allow for these viruses to evolve. So a lively debate and lots of interesting data to support. There's data to support all 3 of these. And I think I'm not a virologist, but it does seem plausible that there might be multiple routes of how the virus did initially evolve. All right. And so please read through that one. It's like a 2-page thing. So then let's see, I'm trying to remember. So we looked at this idea that there are lots of studies going on right now with viruses in the oceans, and they know that there are tons of viruses in the ocean, and they do play a critical role in recycling of nutrients, both in the upper reaches of the ocean, close to the surface and where a lot of the photosynthetic organisms are. There's a lot of recycling of nutrients there with infection by the viruses, but also down in the ocean floor along coral reefs. Viruses are intimately involved in the process of getting nutrients freed up. Once they break open cells and those nutrients come out of the cells, they can get recycled and used by the organism. So the virome idea, all the viruses in a certain ecosystem, the virome is being heavily studied in different ecosystems, including what's going on with humans. And so these 2 slides are just getting at the idea of recycling, how the viruses play a really good role in recycling of minerals and other nutrients, both really in any area of the ocean, above, down deep. And then another idea with regard to viral ecology is that, this, especially in the ocean systems, they've been looking heavily at this idea that it does seem to keep the presence of viruses and their actions of infection and then lysing cells. All of that seems to play an important role in making sure that there's no one species in an ecosystem that dominates. And so we're thinking about the idea of a balanced ecosystem and diversity in these ecosystems. And in general, if you've taken Intro to Ecology, for example, they've probably talked quite a bit about ecosystems being diverse. That seems to be a healthy ecosystem if you have good diversity. And it doesn't really matter which ecosystem you're thinking about. That's the general rule of thumb, that if you have good diversity of species, that's going to allow for a nicely balanced ecosystem and one that will persist. And they think viruses are really important in that process. When you think about how they can control population levels. And so, like I said, a lot of this research is going on in the ocean systems. And I think that, there's really nice data to support this idea. And so from that perspective, viruses are playing a really integral role in our various ecosystems. I'm sure most of you by now have heard about harmful algal blooms in the ocean, and that's another thing that they point to, that you can get harmful algal blooms to practically disappear within a matter of days with viral infections to those algae. And so here they're showing a particular example of this algae, very interesting looking algae getting infected by the virus. And that lower left picture, all the white area is the algal bloom that is occurring due to that algal species. And you can see the swath of white is, that bloom is quite extensive. After the virus completed its life cycle, you really don't see anything. There's a little tiny blip right there, and that's it. So pretty much a complete wipeout of the algal bloom, which is a good thing. Algal blooms can be very dangerous for a lot of different species in that area. It can lead to death of thousands of fish and other organisms. We know that viruses can transfer genes. We looked at that during the gene transfer section. And so this idea of transduction, where they can take snippets of DNA from the first host and potentially get it into another second host, and perhaps that second host can take advantage of it. So that's the transduction process that we talked about. But don't forget, we also talked about nanotubes, and they now know that nanotubes are big enough in diameter where viruses, the phages, can fit through. And there's questions now about the extracellular vesicles that are route as well for viruses to go from one host to another and potentially exchange DNA in that process. So interesting things to think about. This picture that you're looking at is a title to an article that was published where it's a three-way interaction. You have a plant that has a fungal endophyte and the fungus has a virus. And you have to have all three for this plant to survive in its particular habitat. And so in this particular example, they're showing that if you get rid of the virus from the fungus, the plant won't survive. And so they need all three. So here it's the plant has adapted to really hot conditions, and the only way they can survive those hot conditions is if they have the fungus with the virus in that fungus. And so here's another idea where you have these complex interactions amongst different organisms, and you need, in this case, you need the virus in fungus in the plant. So this is trying to get up to 65 degrees Celsius, by the way, is really hot. That's the temperature pretty much where we set for your geobacillus bacteria experiment lab. It melts the Petri plates. I don't know if you saw any of your Petri plates. It's pretty melty. So that's the downside to trying to work with that temperature with Petri plates. Okay, so we know that these double-stranded DNA viruses, when they get into the double-stranded DNA form, they can paste their DNA into the host and just stay there. That viral stretch get integrated into the host DNA. Maybe it will get activated, but they've seen time and time again that it often just stays and becomes part of the host DNA, and it can get handed down into the next generation as just part of the host DNA, that the chromosomes. And so that can happen in the microbial world. What we often see is that some species that cause trouble for us, bacterial pathogens, for example, the only reason they're doing that is because they got infected with a virus who passed them the genes for toxin production. Ordinarily, these species, if they don't have that phage in them, that stretch of DNA, that prophage, they're not going to bother you at all. But if they get that infection, get that prophage state where the virus it pastes its DNA in, then that bacteria becomes pathogenic to them. And then there's also very interesting studies going on with our genome and our chromosomes, looking at the viral components in those, and that they have, this is a, through the evolutionary process of humans, we see that these viral stretches of DNA have been integrated into our chromosome, and they're just part of our chromosomes. And so this particular table, I think, is in your book. And if you kind of split it in half, where the white on the left area, that's all talking about what's going on with bacteria that get DNA inserted in, viral DNA inserted in. So that, remember, prophage, when you hear that word, you're talking about viral DNA, phage DNA, that has been pasted into the bacterial chromosome. And you can see that prophage state allows for Clostridium botulinum to be able to produce the toxin that can kill you in a heartbeat. It takes 0.000005 micrograms to kill a person. So it's an incredibly potent toxin, but if they don't have that prophage state, you can be interacting with Clostridium botulinum just fine. It won't harm you. Diphtheria toxin, that's another serious disease, of course. Shiga toxin, that's also really nasty. Cholera toxin, so a lot of kind of heavy hitters. And it's all because that particular species of bacteria has some of that viral DNA in its chromosome. So that prophage, they're in the prophage state. The pink on the right side of this table is looking at what's going on with humans. And so this is thinking about the idea of our ancestral humans getting viral DNA into their chromosome, and it's just stuck. It's just part of our chromosomes moving forward. And so you can see a couple of these here are looking at placental maturation. So that's a pretty fundamental thing. If you don't have a functioning placenta, you can't give birth. You're not going to be able to progress into the next generation. So obviously, it's super important part of the human process here. You got to have a placenta that work. And so they're finding that ability for our placenta to mature and be able to hold developing fetus, you need that viral DNA. And so it's really kind of astounding to think about it, how intimate the viral DNA has become into our chromosomes, and it's making us who we are, Homo sapiens. And so it's really, when you start diving into this literature, it's really, really kind of mind-bending on what they're looking at and how they're pointing out, the importance and relevance of these viral pieces of DNA and what they've done for us over millions of years. This is from that reading of Trillions Upon Trillions. And so I took a couple of quotes from it. And so, again, most people who are non-biologists, non-microbiologists, they just think of viruses as evil entities. But they're essential to everything from our immune system to our gut microbiome to the ecosystem, land and sea, to climate regulation and the evolution of all species. Viruses contain a vast, diverse array of unknown genes and spread them to other species. An ancient virus that had inserted its DNA in of horned animals that was human ancestors. So way back when, that snippet of genetic code called ARC is part of our nervous system of modern humans and plays a role in human consciousness, nerve communication, memory formation, and higher order thinking. So it's really making us human. And yet it's these structures of viral DNA from way back when that have just become part of our speciation, part of being human. 40% to 80% of the human genome may be linked to ancient viral invasions. Most people have that's not something most people know. And if you told a non-biologist that, they might freak out, but it's not a freaky thing in that regard. I mean, it's an amazing thing, but they're realizing how much viruses have just impacted how fundamental they are to our speciation, our evolution, our ability to function, our ability to persist, not go extinct. Here's, speaking of not going extinct, here's another example of how important viruses are for this parasitoid wasp, where what it does is it will inject its eggs or larvae, we call it eggs, I guess, into the caterpillar, a living caterpillar. So the caterpillar is alive, and it has to be alive in order for this to work with wasp. And it'll inject its eggs, but then it also injects in a bunch of viral particles, and it's got to do both. And then what happens is the viral particles create a situation where the caterpillar's immune system can't fight off and destroy those eggs. And so it's a kind of a sending a little, all these viral helpers along for the egg to persist, be able to mature, grow, and complete its life cycle. So again, an excellent example of how viruses are absolutely necessary for this particular species to continue on and not go extinct in a very interesting way. Okay, let's see. Where's the other? Yeah, we'll do that one. All right, so we hear a lot about viruses causing disease, and there's no doubt that there are some very deadly viruses out there, and we've all lived through a pandemic. And now they're realizing, and when they study these pathogenic viruses, that there are different host ranges. Some viruses are super picky and can only really latch on to certain receptors. Others have a broader host range. And so they often use the example of HIV, very specific, whereas West Nile, that virus, which is present in this area of the country, can certainly affect multiple different species. And this is surprising to a lot of people too, because we always hear about the acute diseases that make you really, really sick and hopefully you recover from. But chronic viral infections that are just kind of low, they don't make you super sick typically, or maybe they do for a little bit, but then they taper off and you recover. But they can stay with you. And so chronic infections, they think, are more common than the acute pathogenic viruses. And one thing that's really lacking is antiviral drugs. We don't really have a good suite of options to pick from when we do, as we all lived through the pandemic. We saw how we just were not prepared for, how to combat it using drugs. And so not till it, had gone on for quite a bit did they come up with something. So that area of research is slow going, unfortunately. There's just not a lot, like compared to antibiotic. We have a lot more antibiotics out there, although we're still running into the problem of antibiotic resistance. But compared to antivirals, we're doing better with the antibiotics, that's for sure. So this has gained intense study compared to just a handful of years ago, looking at especially a human gut microbiome, environment, I should say. And so they're really, really looking at that. And they're pointing to viruses, particularly the phages now, the phages that infect the bacteria that are living in you. That's helping keeping our gut microbes in check, just like we, similar to the coral reef idea or the ocean system, that having these viruses, these phages interacting with our gut microbes is a way to keep population levels in check, make sure there's some diversity going on. And so they're trying to tease this apart and figure out how much phages are playing a role in this process. A lot of the initial research into the human gut microbiome has been a lot of microbe, like bacteria interactions, what's going on there. And now they're starting to realize we got to look at the viral component, the phage component. And so, and they know that phage particles, even though they attack bacteria and get into bacteria, they're actually finding that they can directly interact with our immune system. And so they're not limited to just impacting bacteria. They're realizing that the human host is also able to interact with these phages. And so in this particular case, the study that they were talking about, it was T cell, suppressive T cell activation and tumor formation, which is, you don't want tumors, so that's actually quite interesting. And they're saying that phages could keep biofilms in check, which in this particular paper, that's a good thing. They were looking at biofilm formation in humans. They're saying that the speculating phages are helping with keeping that in balance and not allowing things to get out of whack. So a lot more research going on in the last few years on viral phage, particularly bacteriophages. But looking here is just a kind of a visual, getting at some of these ideas and how you have the viruses playing a role directly with the bacteria themselves. But they're also showing that they can interact with human cells, epithelial cells, and they seem to be influencing some of our immune system regulation. So luckily, we have pretty good technology now to study and ask some pretty interesting questions about our gut microbiome. And the technology is there where we can get some good data and it's not too expensive. It's still somewhat pricey, but it's come way down in price. But some of the research that they're getting into now is just fascinating. So this is a published paper from just a handful of years ago. Sociovirology, conflict, cooperation, and communication among viruses. So now they're starting to ask this question, can we look at viral ecology through the lens of sociology? They already do it with bacteria. There's a whole field of study called sociomicrobiology, where it's primarily focused on bacteria, some fungi work too. But now they're dipping down into the viruses and saying, are viruses social? And the few papers I was reading as I was looking through this, let me see if I can show you. Come on. Am I duplicated? Yeah. Okay, let me show you this. Yeah. Where is it? So this is a Google search on, I just typed in, so, oops, you can't see it. All right, did it to me again. Come on. Okay. Social interactions among bacteriophages. The Great Viral Team Up is another paper that was published not too long ago. The Secret Social Lives of Viruses. So it's a thing. They're starting to apply social evolution theory to, they've already done it to bacteria with pretty good success. So basically taking like how humans interact with each other, how other animals interact, that evolution theory, that social evolution theory, and bringing it down into the microbial realm. And I remember when it first came down into the microbes, primarily the bacteria, and there was a lot of pushback initially. But then they started collecting data and they're like, no, microbes are social. They do talk to each other. And now they're saying, well, oh, what about viruses? And it looks like that is also being taken seriously, that this is not something that's going to be dismissed, and they're getting some good data on it. And so you're going to probably hear more about this, but one of the exciting offshoots to this is that if they can better understand the social interaction of viruses, that can really help with viruses that cause disease in humans and other animals to better understand how to combat them. And just like with the quorum sensing idea, if we can stop them from quorum sensing, they can't really effectively attack you. And so again, with the viruses, could there be something like that where if we could understand how viruses interact? And like I said, I'm not a virologist, but some of the, one of the papers, well, this paper, let me go back into, I'm going to start all over here. Go back here. Is it going to take me there? Okay. So then this paper, here's an excerpt from that paper. Virus-virus interactions are pervasive and highly diverse. Some viruses need another helper virus just to complete their infection cycle. And viral proteins can mix and produce mosaic-like viral particles. So that left-hand paragraph pretty much just kind of demonstrates all the different ways that viruses interact. And then their last paragraph of their intro section is saying, we think you should use a social evolution approach when you're studying viral ecology and looking at these interactions. And we outline how this approach could lead to new breakthroughs in both fundamental and applied virology. So, I mean, when you think about what we just lived through with the COVID pandemic, if they could nail this down better, and when the next pandemic comes, hopefully not in our lifetime, but they predict there's going to be another one somewhere, sometime. Wouldn't it be wonderful to have this kind of solution in place, ready to go? And so I think there's a lot of great potential here. Unfortunately, we have a ways to go, but I think there could be some interesting ways to combat viral infections through this information and learning from. So speaking of diseases and issues, I did want to mention a little bit about this idea, viruses and cancer. So I'm not going to get into huge detail. I think maybe it gets covered a little bit in advanced cell class, but they say roughly 20% of cancers in humans are caused by viruses that are called oncogenic. Onco is referring to cancer, genic is genesis, to create. And so now, obviously that can't happen if the virus kills the cell, because then the cell's dead, it can't become cancerous. We're not talking about the viruses that just obliterate cells. That's not going to lead to cancer, but they know that there are some viruses that can infect a cell, but don't kill the cell. And sometimes when that infection occurs and that cell stays alive, it can lead to a state where that cell becomes cancerous, uncontrolled cell growth, which can lead to tumor formation and cancer. So most viruses don't cause cancer, even the ones that infect us. You know, when you look in those, all of those fatalities, the vast majority don't do that. But so, for example, the cold virus, polio, influenza, all of them, they are not demonstrated to do this. Okay, but I did want to get into there's five right now that are being heavily studied. So what I mean by the whole cell not getting killed is some of these options here. So the one straight across off to the right, that would cause cancer because it's causing cell life. But you might have these other mechanisms play, persistent infection, where the virus is multiplying to a very low level and slowly enough that the cell stays alive and the virus is also replicating. And then you have the transformation idea is often where you get viral DNA pasted into the host chromosome in the wrong spot, which can lead to the gene activation that can lead to tumor formation. So some ideas here of, I'll explain a couple. So I did want you to know the names of these 5 viruses. So hepatitis B and C, human papillomavirus, human T-cell leukemia, Epstein-Barr, and HIV. So those are the big 5 that have been studied. So these viruses can cause cancer. Do they cause cancer all the time? No. So probably I would wager the majority of us in this room probably have EBV, Epstein-Barr virus. If you've had mononucleosis, you have it. It's permanently with you now. I had mono back in high school. So are we all going to get cancer? No. In fact, the EBV type of cancer is limited to certain geographical regions of the world. So they know that there are other influences that's leading to a cancerous state. It has something to do with the environment that those people are living in and their interactions with that environment. So what I'm talking about here is a couple of ideas where here they're showing this idea of the papillomavirus, which can cause cancer, and they're showing the DNA of the virus getting inserted into the host cell. And they're saying that it is being inserted in such a way that it leads to that cell to start to do inappropriate cell division. And so it just depends which stretch of DNA the viral DNA interrupts or pastes into. If it messes with a certain stretch of DNA that controls cell division, and now all of a sudden that's messed up, you can get this uncontrolled cell division, which can lead to tumor formation. And in general, tumors are not, they're your cells, but they don't work for you. They're not. So, for example, if your lung tissue starts to become tumorous, your lung cells no longer help you breathe. They're just taking up space and causing a drain on your resources. And so they're your own cells, but they're just not working for you anymore. And so here, trying to show again, you've seen this idea where if you get that viral DNA piece in the wrong spots in the chromosome, here they're showing prophage, but it can be a provirus, like if they are infecting humans, it can be problematic and often leads to uncontrolled cell growth and tumor formation. So we have these genes on our chromosomes called proto-oncogenes, and they're normal. Everyone has them, right? And they normally promote cell division, which we need in order to grow. We got to have some cell division going on. But if you have a viral DNA segment that gets inserted next to a proto-oncogene, it might cause that proto-oncogene section to get over-transcribed, too many copies of it, and essentially you end up with this ramped up cell division going on, all right? And so fancy way to say that is mitosis is triggered inappropriately. It just means you have kind of uncontrolled cell division going on. And so that they've seen that happen. Another idea is we have tumor suppressor genes, which they do what they say, they suppress tumors. And so through your life, some of your cells become cancerous, but because of the tumor suppressor genes, they can put the brake on that. And you never knew that that was happening because you didn't feel any ill effects. And so this is happening for you as you live your life. But if, again, the viral DNA gets pasted into a section near the tumor suppressor gene, it might cause that gene to not work correctly. And again, you would not be able to have that tumor suppressing function occurring in your body. So these are some of the ideas I'm just trying to get across of how pasting DNA in would be not such a good thing, depending on the sections of DNA that it gets pasted into. The next series of slides are looking at these 5 viruses. I'm not going to ask much about these other than being able to recognize the top 5. And then the big picture idea for most of these viruses is that they can either act directly or indirectly to cause cancer. And so directly means that they're usually interrupting a key gene that's controlling cell division. It's that kind of idea. All right. And then I just give a little bit of detail of these viruses. But I'm not going to ask the details. This is just for your own kind of, if you're curious. All right. So that when I say it acts directly, they usually see that the viral DNA is interacting with the host DNA in such a way to typically cause inflammation or tumor formation. So hepatitis B is a very successful virus from its perspective. It's common worldwide and spread by body fluid contact. The human papilloma, which actually they've done some studies on college campuses across the nation, and it's very successful across the college-age students. It spreads very easily. Benign and malignant warts possible, and cervical cancer. And again, common worldwide, it's all over the place. Every year, 6.2 million people become infected. So this is another one that is messing usually with the gene expression of the host. The human T cell, if you're curious about that one, it's causes T cell leukemia by infecting T lymphocytes. This one's not common worldwide, just key geographic regions. And it's distantly related to HIV. It's a single-stranded positive strand retrovirus. HIV, which most of us have had some exposure to learning about it at some science class. This one we say is an indirect cause of cancer because it's indirect because it kills off so much of our T cells that it hampers our immune system's ability to keep our cells healthy rather than having some of our cells go cancerous. So your immune system's always patrolling and looking for pre-cancerous or cancer cells, and it's able to usually get rid of them, nip that in the bud, and you don't even know that's happening in your body. But when your T cell populations crash, then it doesn't do that for you anymore and can allow for cancer to develop. This is the infection cycle, if you're curious. And the patients are diagnosed with AIDS when, I have in the notes section, when the CD4 T cell count drops below 200 cells per cubic millimeter. So if that's the test result, then they start declaring that you have AIDS. So, and then the Epstein-Barr virus, which causes mononucleosis. So it's very common worldwide, but it doesn't cause cancer worldwide. It's usually only in Africa and China that we see elevated rates of the Burkitt's and other B-cell lymphomas. So those are pretty rare cancer types, but in Africa and China, so it's very common worldwide. Normally, it causes mononucleosis, but just because you have it in you doesn't mean it's going to lead to a cancer. They call it the kissing disease because it needs actually a lot of viral loads. So you typically need quite a bit of kissing for the transfer to be effective. A light peck on the lips isn't going to do it. So, all right. Here's another group that can cause some serious problems, but not in us. Viroids. So this is bizarre. All they are is RNA. So they don't have a capsid. They're not, it's just RNA. That's all it is. And it's a single-stranded. It tends to wind around on itself, but it is very successful at causing plant disease. There have been some terrible wipeouts of plants populations due to a variety of different viroids, but they don't touch animals, only in plants. And so again, really simple, just RNA, but they can be devastating to different, primarily they document this in different crop systems that they're trying to grow for food production. And then another wacky thing, prions, depends who talks about it. I say prions, some people say prions. They're just protein. I'm sure most of you have heard about prions by now, I'm guessing, in some class. I know that they do mention it in some other classes too, but really, really kind of bizarre, where all it is really is the idea that these proteins fold into three-dimensional shape the wrong way. And so the normal folding is being shown here on the left. For the cell and molecular, about alpha helices and beta sheets of protein formation, and you can see visually very different 3D conformation of these proteins, and that makes the difference. And so once you go through translation and make that string of amino acids, that's not really the final protein. Most of those amino acids have to fold in a very particular way in order to function correctly. And so we see with these prions or prions that they don't do that. And so what tends to happen over time is that they stop functioning. They tend to clump together, and it's almost a domino-like impact where they can interact with healthy proteins and cause those proteins to become incorrectly folded. And so there's like this domino effect of these prions interacting even with normal proteins and can cause this kind of chain reaction of failed proteins. And so if you get enough of that happening in one part of your body, you're going to be in trouble. And so that's what they're showing here in figure B, that brain tissue that has had significant prion clumping and damaging going on. Cells start to die, and then you get these blank spots, holes essentially in your brain tissue. And so obviously, if that happens enough, you can see where that's going to lead to significant problems for the person and ultimately death. So the way that it looks under the microscope, where you have all of that tissue damage to complete like gaps of tissue, they call it spongy brain disease, or the fancy word is phrase is transmissible spongiform encephalopathy. What a mouthful. TSE, spongy brain tissue essentially. And so they often refer to the clumped up proteins as plaques. Because they're our own proteins, we don't have an immune reaction. They're our own proteins that get folded in inappropriately. So, like I said, it can lead to severe problems. So these proteins are a little weird because, well, let me finish the illness idea. So the illnesses that can result from prions going wild, Creutzfeldt-Jakob disease, CJD, and there's different ways that this can happen. They say it can be spontaneous, it can be genetically inherited. Iatrogenic just means that you can get it through surgery, like surgical instruments. So that's like in a hospital setting. In fact, I think there's still rules in place that when surgeons perform brain surgery or spinal surgery, they can't reuse the instruments on another person. Even autoclaving, that wouldn't work. It's not safe enough. And so if there's any prions on that instrument, autoclaving is not really going to take care of it. So it's my understanding that they have those laws in place that you have to use brand new equipment for those types of surgeries. Probably one of the more famous examples was in New Guinea, Kuru disease. Can't say that word. The New Guinea population regularly did cannibalism practices as part of honoring their dead. They would dismember the person, and people would eat different parts of the body based on their rank in the society. And some American scientists, I think they were American, some scientists were there interacting with them. I think they finally convinced them not to do that because it was spreading Kuru. And they were able to show the data to demonstrate that that was not good practice. Sheep and goats, you can get scrapie disease. You might have heard that if you're pre-vet. That's again a prion disease. And then chronic wasting disease in white-tailed deer and elk and so on. That's also a prion disease idea. And then probably the most famous is bovine spongiform encephalopathy, which they've nicknamed mad cow disease by the common media. So they talk about mad cow disease from eating contaminated meat that was inappropriately butchered. And so now there's new laws in place for butchers on how they can cut certain parts of the cattle meat and how they have to avoid certain nerve areas and of course the brain. And so technically the new variant CJD is the human form of mad cow disease. So if you were to eat infected cow meat, essentially, they would call it the new variant CJD. So problem with this, well, one of the many problems with this disease is you don't know you have it because it can just kind of be in you doing its thing for a couple of decades before it starts to manifest any serious issues. And another bizarre thing is that they're proteins. So you'd think, I mean, we learn in basic science classes that, well, if you just heat proteins, they should denature and stop doing what they're doing. Not with these. You can kit them with all kinds of things that normally work to disintegrate proteins out of their, three-dimensional state, and they can survive it. And they hit it with radiation, all kinds of disinfectants, detergents. So I have in the notes that one paper showed you can denature and deactivate them by subjecting them to a temperature of 134 degrees Celsius, which is hotter than autoclave, for 18 minutes. So you have to up the autoclave more than what we usually do, 121 degrees. This is 134, so you have to bump it up in temperature and pressure. And then another paper I read, it said you can use bleach, but it says you need to expose the prion to 1 hour of the bleach. And so most people, when they disinfect a surface, they're not going to do that. So that's another problem. All right. This is just a visual trying to show the chain reaction of the prion protein. And this is scrapie and sheep and goats, and how they're trying to show it's more of a crescent shape. A normal prion protein is not flat shape. And they're trying to show that when they interact, they slowly start to create this chain reaction of incorrectly folded proteins. So, we had these proteins are just part of us, and it's one of our own proteins that's kind of gone off the grid. Weird thing. This is showing this idea that we have normal prion proteins that function just fine, but it can become abnormally. So those are just trying to show that same idea of abnormal formation and causing problems, including spongy brain tissue. Okay. All right. We'll stop there. Have a good weekend. And the next topic we're getting into is the human microbe interactions. We'll start that next week.